So we make the blood smear in slides like this, right? So do you have any idea why we make this blood smear? Sir has already given you some insights on it. Yeah, tell me. To view the different components of the blood. Okay, blood cells, right? There are many blood cells like RBCs, granulocytes, and agranulocytes. So, based on the chemical structure of the chemical composition and the morphology of the cell, we can view various kinds of cells in a smear. Okay.
We have to identify the blood cells. What are the
Huh.
different blood cells?
Bottom line is we have to identify all the blood cells. Okay.
And you have to find out the percentage of different cells.
Yeah, that will do in differential leukocyte count. Okay. For that, you will need a smear again. Next is we need to stain. We need to put a proper stain on that smear because without stain, nothing will be visible. Okay, everything will be alike. You won't be able to see various cells. Okay, so some components of the cell are acidic and some are basic, right? So the acidic components are negatively charged. We know that acids are hydrogen ion acceptors and the bases are the H plus donors. So they are positively charged. Okay, so one component of the stain stains the acidic particles and another component stains the basic particles. Based on the chemical composition of the cell, thus we identify various kinds. Okay, so the stain that we use to stain is the Leishman stain. Okay, spelling is Leishman, Leishman stain. So it belongs to a group of class of stain which is called Romanowski. Romanowski. Okay, so it has got three components in it. First is the eosin, eosin, which is the acidic component of the dye. So the acidic component will stain all the basic parts of the cell.
The acidic component will stay in the basic part of the
Acidic dye.
Can you tell us which component of the cell is basic?
Anyone please tell us? What do you think? Which is basic? Eosin is the acidic dye that will stain eosinophils. Okay, yeah, it will stain eosinophils. So, cytoplasm of the eosinophil is basic, and RBCs are basic. Okay, and another is the methylene blue, which is the basic dye. So, accordingly, tell me the basic parts of a cell. So, what are the acidic parts? I have told you the basic components.
Nucleus.
Basic is
Think about nucleus. Whether it is acidic or basic. Acid. So which stain it will take? Means which stain?
basic stain. Name of the basic stain.
One is acidic component and another is basic component. Basic component is methylene group. So whether how we will be staining the nucleus? Which methylene group, right? And the cytoplasm and cytoplasm proteins, no, it is granulocytes. So these are
Granules of basophils.
huh? Proteins are acidic or basic. Basic. Negatively charged. Mainly proteins are negatively charged. At the pH, cellular pH is 7.4 also. So at this pH, proteins are negatively charged. The component which are negatively charged, what we call them? Which are
Acidic.
basic.
So
Those
so negatively charged is basic and positive.
basic, this is the basic component. So actually these are basic. So they will take acidic dye means you see. Means they will take eosin. They can be colored by the eosin. And in the cytoplasm, some of the cytoplasmic granules, they take acidic state. Some take basic state. Some granules take of some cells they take both the state. Means they are neutral. They take neutral states. So accordingly, they will produce those who will be taking acidic state. Their color will be different from the stain from the component which are taking basic state. Similarly, those who will be taking both the state means neutral state, their appearance will also be, their color will also be different.
Okay.
So the granules also will vary.
Granules of basophils will be stained by methylene blue. Okay.
Can I ask
Methylene blue is a basic dye and granules of basophils are acidic. So that will stain it.
I will ask one more question. Two. Lymphocytes. In lymphocyte, what type of granules you will find? You don't know? Okay. Can you tell? Lymphocytes, whether these lymphocytes will contain granules or not. There will be granules. Correct. The lymphocytes are agranulocytes. Another cell is there. Monocytes. So these two are agranulocytes. Others are granulocytes. So they are of three types. No. Three varieties. One is eosinophil, one is basophil. Okay? So you have to identify all those things.
Yes.
That's why name is given.
Eosinophil because stained by eosin, and basophil stained by methylene blue. Granules are stained basically. So the third component is acetone-free methyl alcohol. Okay, methyl alcohol preserves the cell in whatever chemical and metabolic condition they are in. Okay, and we have to keep the methyl alcohol free from acetone because acetone causes crenation or shrinkage of the cells. Okay, is that clear to you? The components of the Leishman stain. Do you have any doubt about it? Okay, can you go to the smear making proper, right?
Okay. Now we will go for, you told about the material
Leishman.
required.
Yes. Apparatus, I will tell
What are
sir.
the materials required? Slides. You will be requiring spirit also. If your blood is given in a vial, then how will you take the blood from the vial? You also need a vial, a dropper also. And do you need a cotton for cleaning? Remember one thing. So let's see the procedure. Okay. So start with the procedure.
So the procedure, now we'll learn about it, okay. We'll take 4 glass slides. Ideally, we should make 3 smears, okay. But for practice, we'll make only 1 now. So we have to take 2 slides for that, okay. So under all,
One of the slide, it is only for the purpose of
making the smear, spreader, it will be used as a spreader.
is you select the spreader first, and you just have to see there whether the surface is smooth. It should be smooth. Okay. So this is one requirement for making a good smear.
So see here we have taken 2 slides, right? This one will be used as a spreader, okay? Firstly, the slide should be clean enough. There should not be any grease, okay? And then under all aseptic condition, you will prick your finger again, like you used to do all the time. Sir, okay. So 1 to 2 centimeter from this end, we'll just touch the drop of blood with it, okay? After that, we'll place the spreader here. Okay, see sir is showing how the spreader is placed at an angle of 45 degree. Okay, 1 to 2 centimeter away from one end. Okay, then we'll place the spreader here at an angle of 45 degree. Should not be more or less than 45. Okay, otherwise your smear will be erroneous.
This is blood. So you bring it closer to the blood. Now the blood will spread along the surface at the joint. So when it's spread, then only at 45 degree angle. So this is 90 degree. Half of this is 45 degree. So then I will push it forward. Not so fast, not too slow, not by zapping. This is not the way. It should be smooth. You should keep the slide on the surface. Okay?
Yeah. See, we will place the spreader here and then we will move it backward. And the blood drop will spread along this width. Okay? Then along an angle of 45 degree, we'll spread the spread the blood. And the spreading should be smooth enough. Okay? You should not be hesitant while spreading the blood. After that, we'll allow the smear to dry in air. We'll leave it. Okay? After that, our staining process will start. Now, see how an ideal blood smear should look like. Okay?
Which one is ideal?
There are some criteria. Can you tell us about the criteria of a good blood smear? Okay, is it clear? An ideal blood smear, it should be tongue shaped ideally. 3 to 4 centimeter should be its length. Should not be too thick or too thin. If your angle is lesser than 45 degree, chances are there the blood smear will be too thick. If it is more than 45 degree, then it will be too thin. And no vacuoles should be present. See, the vacuoles are present, this should be absent in an ideal smear. Okay, you have to learn the criteria about it. Okay, so next is staining. Now we'll start staining the smear. Procedure is written here. Okay, any one of you come and read the procedure once. Yeah, Madhurima, you come.
Uh, Preparation of a blood smear. The first step is take 4 or 5 clean grease-free glass slides with smooth edges, 3 to 4 to be covered with the blood film and 1 to be used as a spreader. The second step is lay 3 or 4 glass slides on a table, prick the finger, touch the bleeding point lightly in the center line, about 1 or 2 centimeters from one end of the slides to obtain a small drop of blood on each slide. The third step is place the narrow edge of the spreader on the surface of slide just in front of the blood drop at an angle of 45 degrees. Draw the spreader backwards so as to touch the blood drop and hold there till the blood runs along the full width of the spreader at the line of junction. And the fourth step is the spreader is then moved slowly and smoothly to the other end of the slide, maintaining an angle of 45 degree. The fifth step is allow the film to dry in air. Repeat the procedure with other slide.
Okay, so you have to now fix the smear and stain the smear. Fixing the blood smear. Okay, now we'll place the blood smear across. We'll put it in a support. Our slide, we'll put it in a support and then we'll start staining it. Now it's written that Leishman stain that I have mentioned about, we have to put 8 to 12 drops of Leishman stain over the slide and cover the slide. Cover your smear with the Leishman stain and leave it that for 2 minutes. Within 2 minutes, the cells will get fixed in their position in whatever chemical and metabolic state they are in. Okay, we have to give 2 minutes time for that. So fixation time is 2 minutes. Okay, this is the support on which you will place your slides. Okay, so next is we have to note how many drops of Leishman stain we have poured. Suppose we have poured 8 drops of Leishman stain and we have covered the entire smear. So after 2 minutes, we have to add an equal amount of buffered water. What is a buffered water? Buffered water is maintaining the pH level at exactly 6.8. Okay, if we are using buffered water, if it is available, then we will put equal number of buffered water on it. Okay, if buffered water is not available, we will use distilled water and we'll double the number. Suppose we are putting 8 drops, then for distilled water, we will use 16 drops of it. Okay, so you have to calculate the amount of Leishman stain you are putting on it. Okay, see it is written over here. Next is we have to mix the stain and the water. For that, we'll take a dropper and we'll blow on the dropper, or we'll press the dropper and the water and the stain will get mixed, okay, evenly. Now, if the dilution of the stain is correct, you will see a greenish layer or a greenish scum will form on top of your smear, okay. On the stain and the water that is mixed, a greenish layer will form over it. That means your dilution is perfect, okay. Now, next step is we have to leave the stain for 10 minutes. This is the staining time.
How many drops of Leishman stain you will be giving? Okay. Then how much water? How much time you need?
And stay that we should not place it directly under the tap water, otherwise your smear will get destroyed. Okay, so we have to keep it in a slanting position.
So the flow of water in the tap should be slowly flowing, otherwise wash it.
Okay, we have to wash it for around 2 minutes. After 2 minutes, you will see a pinkish tinge on your smear. So that means your smear is now ready to be visualized under a microscope.
At the time of washing, at the bottom of the, here is the film and some stain may go there.
We'll keep it in air to get dried, and then we'll see it, observe it under a microscope. So this is the procedure. Is it ready to be observed under a microscope?
And the problem is,
The microscope.
we are unable to use the microscope and how to focus it, we are not knowing. So we will be learning this. So since number of microscope are less, so I hopefully we will be getting soon. Then only everyone can be provided with one microscope.